delighted to be joined. Oh, I'm going to read from my sheet, not around the screen. We've got Imogen Swift, who's a research assistant at the UK Dementia Research Institute at UCL. Hello, Imogen. Hello. Hello. We've got Dr. Lucy Russell, who's a research fellow at the Dementia Research. Do you all work really at the same place? Because some of you said, like, I'm in the Institute of Neurology, somebody else said I'm at the DRI, somebody else said I'm at the Dementia Research Centre. But so do you all work within the Dementia Research Institute, which is part of the Institute of Neurology? No, it's not part of the Institute.
It's complex. We all work together. We all work together. At
University College London.
Yeah.
Well done. And are you all funded as part of the UK Dementia Research Institute?
No.
Some are. But that goes to show how interesting and varied the DRI is, in so much as the way it delivers its care crosses over. Its care, its work. So we've got Dr. Martina Bacchetta, who's a senior research fellow at also UCL. We've got Mika Clark, PhD student from UCL. Somebody earlier said, well done for having PhD students on. Like they probably don't know anything, but we've got them anyway. You got PhD students are brilliant. They're loads more interesting than everybody else. So thank you, Mika, for joining as well, and Atina
Aitana Sogor.
Yeah.
Sorry,
Aitana,
my name is not easy.
We've got Aitana. Hi, Aitana. I love you've got planets on your top.
Yeah.
That's cool. Thanks very much, everybody, for joining us. And so the session coming up is all about frontal temporal dementia. And that's because UCL is kind of, are you going to argue that you're the best place, the best, most focused place in the country or even the world for FTD? Is that the case you would make?
Well, we lead.
You're all too modest.
We lead an international frontotemporal dementia project. So it's coordinated at UCL, but we work with many sites across the UK, Europe and Canada. And the difficulty with frontotemporal dementia is that it is quite rare. So by collaborating with all these different research institutes, it allows us to gain more data and gain a better understanding of what is actually going on. Because currently we don't have any treatments for FTD. So the main bulk of our work is to move towards helping pharmaceutical companies to develop treatments.
Brilliant. Well, so you jumped ahead of me there, because you're the only group that seems to have actually coordinated with each other, and you've planned ahead, haven't you? You've decided who's going to talk on what, which is great, because then I can put up my little "I'll be back in a minute" moment and eat something crunchy that I don't want to eat in front of everybody else. So in the order we've got down here, I got was that you, Lucy? I'm going to come to you first to ask you to introduce FTD. Yeah. Your research projects, clinical trials, and your support groups. Is that what you've just done?
No, I can give a little bit. No, I can definitely do more.
Okay, great. So, Lucy, why don't you give us the you do your bit of the first.
Sure. So FTD is a rare form of dementia. It affects individuals' behaviour and their language instead of their memory, like you have in Alzheimer's disease. And we don't really know much about it at the moment. So we've got 2 longitudinal cohort studies. So that means we see participants over a long period of time, over many years, to try and find out how the illness progresses and who it affects and why it may affect them. And these 2 studies we have are, the first one's the longitudinal investigation of frontotemporal dementia, also known as LIFT. Now this project follows people who we don't know why they have frontotemporal dementia. And the second one is the Genetic Frontotemporal Dementia Initiative, also known as GENFI. And this follows individuals who have a family history of FTD. So that means that either their parents or their grandparents have had frontotemporal dementia, and it means that their children are at risk of developing FTD at some time in their life, so a 50% chance that they will develop it. But this gives us a really valuable opportunity to follow these people before they develop symptoms. So we can see at what age they may develop symptoms and it gives us a window into a period of time that we often miss in research, where the symptoms are very mild, and it's not until later on, where the symptoms are becoming very problematic in everyday life, that they present at clinic. And this period is really important for clinical trials. If we want to treat this form of dementia, it might be better that we start treating it at the earliest stage possible. So the work with my colleagues here today are going to talk to you about how we're trying to do that. We've got various measures that we use in these projects to assess these things, and we are making progress. We currently have one clinical trial up and running. It's an international trial, but we're also running it at UCL, looking at a particular form of FTD, a familial form that's passed down, and we've got a couple of other clinical trials that are in the pipeline to be rolled out within the next 2 years or so. And this is the main focus of our work, to find out how we can move towards these therapeutic treatments.
And your study participants, so you're obviously, as we said, you're at UCL, which is in London, but your study participants don't all just come from your hospital in your area, do they?
No, not at all. We have people from all across the country. We have people coming from Germany, if they're based in the UK, but currently living overseas. We also have people from Ireland coming over. So we like to see people from across the country if we can.
And you, when you talked before about the GENFI study, so people who carry this particular gene are 50%, there's a 50-50 chance that they'll go on to develop that particular.
Yeah, that's right. So FTD is extremely complex because there's multiple genetic factors and different genes that can cause FTD. So we're not just treating one illness, there's multiple and multiple causes, which makes it very difficult to develop treatments. And I think Imogen and Atana are going to talk a little bit more about the genetics of FTD a little bit later.
Absolutely. Well, that leads us quite nicely into Martina's, because that's the, I think you're next on my list here to talk about the genetic initiative. And can you tell us about brain anatomy, MRI?
Yes, I will. So first of all, I'm funded by Alzheimer's Society, who is one of the greatest charity who are doing lots of amazing work to help and support research to help people living with dementia. So as Lucy said, we don't really know what is causing frontotemporal dementia, but what we know is that the only known risk is genetic. And about 30% of people living with frontotemporal dementia do have a mutation. What it means is that they are born with this problem in their genes, and at some point in their life, they will develop cognitive or clinical symptoms. So someone might develop some changes in behavior, some others might develop problems or difficulties in language. And the problem is that we don't really know why some people might develop one form or one type of symptoms and why others may develop different things. But what we know is that the reason why it's so variable is because different parts of the brain are affected. So we tend to consider the brain as just one single thing, but actually it's not. So the brain is formed by different areas and different structures, and each of these structures have a specific role. So for instance, if someone starts developing problems in their language, it means that probably there is some pathological processes going on in the area that has a link with language. Or if they start developing some symptoms linked to changes in behavior or problems in showing their emotion, then there is another part of the brain that is affected. So what is possible to investigate in people with a mutation is to look at years before the onset of these symptoms. And it's possible to measure what is going on in their brain. So we can try to understand and link what is going on in the brain and link with the type of symptoms that they will develop further on.
So, I mean, is that right? So when you said before, the only known risk of FTD is this genetic one. So some of the other ones that they've been identifying for Alzheimer's, for example, some like the gene, around sleep and things like that. So those don't, I mean, clearly nobody's not going to not act on those modifier risk factors because they might affect your Alzheimer's, but they're not known to also affect FTD.
No, so there are things that might kind of suggest to be linked with development of dementia, frontotemporal dementia, but the only clear cause is genetic.
And can you have FTD and another type of, you know, when people have talked before about mixed dementia, could you have FTD with something else as well?
You have to be really unlucky, but yes, it's possible.
I think one of the biggest connections between two diseases is frontotemporal dementia and motor neuron disease. So in about 15% of people who have frontotemporal dementia, they will also have symptoms of motor neuron disease as well. The combination is linked at the genetic level. So some people will have a certain genetic mutation which will cause both diseases, but it's also the pathology. So by pathology, we mean what's going on in the brain itself. So what proteins are showing problems in the brain. And that's the biggest known link. And there are some other motor diseases, we call them, so that affect mainly the body rather than just the mind or cognition or thinking alone. But they are rarer than just FTD.
Oh, well, motor neuron disease can hit people very early in life. Is FTD then the same?
That's a good question. In very rare cases, there have been patients that show symptoms at much lower ages, but that's incredibly rare as well. So while frontotemporal dementia is the second most common early onset dementia, so by early onset we mean before the age of 65 normally, it's the second most common form, and very rarely it might affect people as young as their 30s or 40s, but most of the people we see are between the ages of around 50 to 65.
And are NHS memory clinics and services good at spotting this? I mean, are they, and referring, because is FTD still, would that still go through that kind of psychiatrist care pathway of, to a memory clinic, you see a consultant psychiatrist and psychologists and you get diagnosed in that way? Did they diagnose in the same, or do you suddenly find yourself in neurology?
Well, I think that's a really good question, because we'll talk a bit about what the symptoms look like in FTD, but a huge part of those symptoms are behavioural symptoms, and typically people don't associate problems with your behaviour as having a form of dementia, and rather as having a form of psychiatric illness. So you will hear stories of lots of people with frontotemporal dementia have gone through quite a tricky path to get to a diagnosis. Often as they might go to psychiatric services for mental health problems before they're identified as having a degenerative disease, which means essentially a dementia.
Parkinson's and things as well. Sorry, did I jump ruin the flow of questions?
No, not at all. We're not rigid.
Well, we've got a moment there. Just a reminder to anybody, if you do have your questions, you can post them. You have to post them as comments in YouTube, or you can use the Q&A button in the Zoom live stream as well, or I will have a look on Twitter at #Chatathon. So let's did you add anything, are we coming to, did you have anything to add, Martina, or do I?
If you want, I can talk about what is the aim and the plan for my fellowship.
Oh yeah, do that. Let's listen.
Okay, so I'm trying to measure what is happening in the brain of people with a mutation for FTD. And in order to do that, I'm aiming to use magnetic resonance imaging, which is a not invasive methodology, which help us to have kind of pictures of the brain. So we can have three-dimensional pictures or in black and white. And it's really, really useful because it gives us lots of information. For instance, we can measure which part of the brain are affected by loss of neurons, if there is something that is widespread, if it's localized just in one area. And we can also measure if two regions of the brain are communicating well to each other, or if there is a problem in the connection of this. If we look at several follow-up visits, we also can measure if there is a change in progression. So if there is a fast or slow rate of progression and atrophy in the brain. So what I'm planning to do is to accurately measure where we can start seeing changes in the brain, when this is happening, and how fast this is going. And I think this is really important because we don't know yet a clear picture of what is going on in the brain. If we don't have a specific and very accurate way to measure that, then we will have two problems. First of all, we cannot tell individuals with a mutation on what to expect in the future. So we need to understand and make a clear description of what is going on. And also the ability of designing clinical trials will be difficult and challenging. So the idea is to have a clear description of what is the pattern and the normal progression of what is going on in the brain of these individuals, because then we can have sort of a reference point to see whether a drug is working or not, whether the drug is kind of slowing down the progression of what is considered to be the normal evolution in the brain. And I have to say that I will be able to do that really thanks to Alzheimer's Society, who is supporting my research, and really thanks to the GENFI initiative, because this is a fantastic initiative bringing together 27 centers around the world. And it started in 2011, so we have several visits, and we really need to thank all the participants and their family to contribute and give them their time to help us in trying to find and understand FTD better.
And is there a particular, so anybody who's watching who has a family member or knows somebody with FTD, is there a particular place you'd like to signpost them to get more involved in research? Because that comes up quite a lot. I'm telling everybody to sign up to join Dementia Research at the moment, but I know the numbers of people with FTD on there are relatively small. Where would you, are we sending them to your new, because you've got a new website, right?
So,
Am I jumping ahead again? Should I? I'm gonna move on. I'm gonna come to and we'll come back to those questions afterwards. So I'm gonna move on to Mika. You're gonna talk next.
Hi there. So my name's Misha. Sorry, I hate to correct pronunciation, but there's never a good time.
Do you know what? I'm getting well known for mispronouncing people's names. Okay, Misha. Right now.
So yeah, I'm a PhD student as part of the same lab as all of these guys, and I work mainly on a different type of brain imaging to understand changes in the brain. Essentially what we do is take the images that Martina looks at and we add colour. So the type of imaging we look at is called PET imaging. That stands for positron emission tomography. But in short, what we do with PET imaging is we give participants an injection prior to the scan, and then they enter a similar type of scanner to an MRI scanner. And this allows us to look at proteins inside the brain. So instead of just looking at brain structure, we look at brain function as well, and how proteins are different in people with a diagnosis of frontotemporal dementia in our case, compared to healthy people. So the kinds of proteins that we're looking at the moment are proteins involved in inflammation. So much like when you have an injury anywhere else on the body and your body has an inflammatory response, so cells that try and help out that site of injury, we think that perhaps the same thing happens in the brain with dementia. So we want to understand that a bit better. And we also look at other types of PET imaging that allow us to look at the connections between brain cells as well. And PET imaging is very valuable because it's the only way to look inside the brain at what's happening in kind of real time, if you like. And that will be hopefully very valuable for clinical trials going forward to see how disease is progressing or hopefully not progressing with treatments as they emerge.
And I had a question then, which I'm just trying to recall. So if you image somebody who, well, first of all, how can you be certain somebody has FTD as opposed to something else?
Well, I guess the real crux of the issue we have at the moment is we can't, unfortunately, until post-mortem, which is essentially until the brain is analysed at autopsy. And that's the only way we do it at the moment. We're developing lots of different markers, which Imogen and Aitana will talk about in blood or other tissue that we want to look at during life to make a more certain diagnosis. But the only way we know at the moment is if someone has a genetic form, and then we can tell exactly what type of disease they have.
So you can do the genetic test and then combine that with what they can tell you about their symptoms, which makes you be able to. So the PET imaging itself you can't see from a PET that somebody has this, but you can see what's going on in the brain with people who you previously.
Yeah, I mean, as it gets more advanced, hopefully we will be able to, because we'll have what we call tracers, so these injections that allow us to look at very specific proteins that are only involved in certain types of disease. So one of the proteins you might have heard about in earlier talks today is tau, and the tau protein is the protein that's gone wrong in quite a few forms of dementia, including frontotemporal dementia. And I think as time goes on and we get a better tracer to look at that with PET imaging, hopefully we can be more sure of what we're looking at in the brain. But at the moment, it's mainly done by where in the brain the problem is. So if it's in those frontal and temporal regions, which is where the name frontotemporal dementia comes from, that gives us a clue as to what disease we're looking at.
And so because this is happening in a particular part of the brain, then are there some unique symptoms associated with FTD over other types of dementia?
Yes, and that leads me on very nicely to the cognitive section. So cognitive markers are essentially the symptoms we see, so the problems in thinking that we see in frontotemporal dementia. And as Lucy mentioned, frontotemporal dementia is very mixed, if you like. We see lots of different symptoms. The main symptoms are either language symptoms or behaviour symptoms, but they're very diverse. So we, in our research studies, do lots of psychology tests, which allow us to look at different aspects of thinking. So language tests might include naming tests. And if somebody's got a problem with naming, we say that's a problem with semantic knowledge. And that might give us a clue as to what type of FTD they have. We do memory tests, so recognition tests, and then we do behavioural tests as well. This looks at a person's ability to inhibit certain responses, so to hold back certain behaviours and responses, because one of the biggest symptoms that we see in FTD, which is a kind of hallmark symptom of FTD or behavioural variant FTD, is when they have trouble inhibiting certain behaviours that you might expect someone to inhibit in everyday life. So things that might be deemed not harmful but inappropriate. And
so this is saying something out of place, or is it? It's not a motor thing. It's not doing something. It's not kind of a glitch in so much as suddenly throwing out your arms or something. Or is it that? Or is it just?
Interestingly, they can they can show some kind of motor behaviours like tapping or repeating actions with certain things. But typically we look at behaviours in things they say or things they don't say in certain situations. And a huge element of that is how they behave socially. So what we call social cognition. And actually, Lucy's done a lot of research in social cognition in FTD.
So typically they find it very difficult to process others' emotions. So, you know, if you were feeling happy and smiling, they may have real difficulty interpreting what you mean by that behaviour. Or as another example, they'd be very socially distant. So they themselves show very few social emotions. And so it can be very hard to interact with them because they're not giving off any social cues that we use in everyday life to make conversations happen or allow us to, you know, just be around one another, even if it's without speaking. Those social, physical cues that we give off, they don't tend to have. And then the other side of it is their ability to be able to think what other people are thinking. So we have a couple of tasks that look at this, and you know, if there's a social situation that they're meant to do something as a response or understand that somebody's done something wrong, they may completely miss that, and of course then their own behaviour is inappropriately placed.
And I guess these, I mean, are these things subtle changes that might happen over time, or do they happen quite quickly? Because from what you've said, if the differences are that significant, you'd expect that diagnosing this would be easy, and it's not, right? I mean, people don't get diagnosed easily, so these must be quite subtle or hard to spot, which means you need rather sensitive tests.
Yeah, that's exactly right. And like Nisha said earlier, they often present as other conditions, particularly depression or anxiety, where people become very withdrawn from the environment. But it does happen gradually over time. And currently, a lot of the tasks that we have aren't sensitive enough to detect these changes. And so we are trying to develop tests that are more sensitive to these changes prior to an official diagnosis, to help us with the clinical trials. And I think we'll touch on that right at the very end. We've got something.
Yeah, and disseminating those, because it's great if these can all be done at UCL, but of course not everybody's going to be at UCL. Yeah, and we are starting to get a nice little, quite a lot of questions coming in for you guys compared to everybody else. So I am going to go to you next, Imogen.
Hi, so I thought I was going to talk a little bit about a day in the life of kind of a dementia researcher, because my job is, well, I do quite a lot of little things. I actually work at 4 different buildings, which proves difficult to walk between them, which makes sense with the whole DRI versus DRC, all these complicated things. But one of the key parts of my role is actually to do with samples, and that is kind of blood samples or spinal fluid samples, which I know you've mentioned previously a little bit, and also urine samples. So these are really useful for loads of different things. Tana's going to talk a little bit about how we use them for fluid biomarkers. But another really useful thing, for example, is that for the genetic side, we use blood to actually work out someone's genetic code and whether they have these genes or these issues in the genes that cause FTD. So as we've said, genetics is obviously a huge part of the disease, and it's something that we really want to work out more about, so we actually know kind of the different types of disease, because as you know, it's super complicated. But also each person has a very different form of symptoms and a different genetic code, and so ideally we can give a kind of personalized treatment at the end of it. So that's a really important part. So when someone comes in for a visit, these amazing participants come from all over the country, and it's really great that they can come and visit us, and they take a blood sample and a spinal fluid sample, and they give us a urine sample. We can then use those samples in the lab, and we can sequence them through our amazing lab at UCL as well, and we can work out individual things about that. So that's a really useful thing. Another really useful part of the samples is for spinal fluid. So I know there's been some discussions earlier in the chatathon about spinal fluid and how it can be quite invasive. The good thing is that it's quite a kind of common clinical procedure, and it's super useful for us because it is a window into the brain, because it's literally the fluid that your brain sits in. So super handy. Also, the blood is super useful to combine them together. So if we can work out what goes on between the brain and the blood in this disease, it can support all the psychology stuff and the imaging stuff as well, and we can create a really great picture of what's going on. So we're really grateful to everyone who helps out with that.
I think people just want you to find a mechanism of getting that doesn't involve a needle.
I can understand that, because it can be quite daunting, I imagine. The good thing is that all the people that I've met who have done it are amazing doctors, and full trust that they will do it amazingly.
Have you all had one? Anybody?
I haven't yet. I'm quite tempted to be honest. I'm very intrigued about what goes on in my spinal fluid.
So I have, only because I donated to the Anthony Nolan Trust,
amazing,
for the bone, and they collect as part of that to put you on their bank. So yeah I know what it's like. Although I am also I'm old enough to be in the Prevent study, which Craig Ritchie's Prevent study, which is probably being talked about right now. I should have mentioned, I think their conference finishes shortly. There's a conference going on in Scotland today. But I didn't, having given to the Nolan Trust, I then didn't want to do it again.
Okay. Well, yeah, I do understand.
So all your patients come to you would have come through a specialist referral pathway. So I'm just trying to get my head round, for those that are watching, somebody who's living in Bedford right now goes through this diagnosis pathway. Maybe their psychiatrist thinks, you might have FTD, but then they probably just get sent home, right? I mean, I'm being told you might have FTD. How does somebody end up at UCL getting this amazing treatment and this chance to participate in things that you do? How does that happen?
I think there are many different routes. I'm sure kind of Lucy can go into a bit more detail, but basically people come from all different kind of routes. So you can go straight to the clinic and you get referred to, for example, Jon Rohrer, who's our boss, and he can then tell you about the research. But there's loads of amazing scientists and researchers and clinicians out there. There's also amazing websites like our FTD Talk website. So I think it's knowing where to look and having those good clinicians to tell you. But
yeah, I think if an individual's gone into their local memory clinic, they've been diagnosed with FTD, there's one of two ways they can go about being referred to us or coming to see us. So that's first off by the neurologist or the psychiatrist that they've seen. They can send a formal referral to the National Hospital for Neurology and Neurosurgery and the Cognitive Clinic. One of the neurologists there will be very happy to see them and refer them into our research projects. The other way is, I think it's still active, but we have the Dementia Research Centre has a research email account, and they can personally send us an email saying that they're interested in participating in our work, and they can come via that route as well.
I think that's the point, isn't it? That this is to participate in research, because we haven't talked about this, but just like other forms of dementia, there isn't currently a treatment for FTD.
No. So this is all cohort studies, as Lucy mentioned earlier, which are a way of following people over time. Normally people enter the study and stay for a couple of years in the study, and we see them once a year. And this really gives us a good way of knowing how things change over time. And of course, an important part of that as well is having healthy people in our research as well. So we have people who are the same age, and I'd say the reason we haven't had the lumbar punctures ourselves is because we don't qualify yet. Yeah, we're too young. But we have lots of healthy people come in and have the exact same procedures as somebody with FTD would. And this gives us a way of knowing really what's abnormal, because you'd never know until you have something to compare to.
Of course.
And just touching on the healthy controls there, we actually recruit our controls from Joint Dementia Research platform. So please do sign up if you're interested.
So did somebody mention before that there is you have got one trial running right now?
Yeah, so we have a clinical trial running at UCL and internationally it's running, and that is looking at a type of frontotemporal dementia that's caused by the gene progranulin. And at the minute, it's still in early stages. We're just coming to the end of the phase 2. All seems to be going well. And hopefully we'll know a little bit more about whether the phase 3 trial is going ahead in the next 6 months.
Yeah, I know early drug trials are always so difficult. So I'm not going to pin you down and start asking if it really works. Otherwise, you'll certainly get a wave of people. We have got a lot of questions. So Aitana, I'm going to come to you next. Could you tell us your bit so I can then get to all my questions?
Yeah, so, well, I'm the one that takes care of the last part, the last piece of the puzzle here is the fluid biomarker.
So Imogen collects, you don't collect it, but you go chat to the person and keep them company while somebody collects it. Or do you do?
Yeah, no, we receive samples.
Imogen collects.
No.
You deal with it.
Yeah, so we receive the samples, Imogen processes them. And well, the focus of this project, our research, is to find fluid biomarkers for the diagnosis of FTD. And well, I think it was mentioned before, but the biomarker is basically something that we can measure in the body and can tell us what is happening, in our case, in the brain. So biomarker can be imaging biomarker, as Misha and Martina were saying before. But in our case, it's fluid biomarkers, because it's something that we measure in human biofluid with cerebrospinal fluid or blood. And these are mainly proteins, but we can also measure lipids. And we focus on those that are changing in FTD. So the thing is that, as all of them say before, FTD is a very complex disease or pathology. And our focus is to try to find specific proteins or lipids that tell us that the person has this condition. So the process is a very, very, very long way. So first of all, we need to find on the literature about the mechanisms that underlie in the brain, and then we select candidates that will be possible to measure and can tell us something that is happening in the brain. And then the next step will be to develop what we call assays to measure them in blood or CSF. And an assay is a test in which you can measure the quantity of this molecule in the sample. And then once we come up with an assay that is able to measure that, we need to validate it for a diagnosis of the disease. And that means is that we have to measure it in samples from persons with FTD, but also controls, but also other disease or other dementias. So we can make sure that this protein is specific of FTD and can serve as a diagnosis for it.
Wow, you're so busy, and we should add that, I mean, you're just a handful of a much larger team as well, aren't you? I mean, I've seen the pictures with Jon's. There are so many of you there working in so many different ways, which is why I jokingly talked about you at the start about this. I know there is working FTD going on elsewhere in the country, but the UCL is definitely got lots going on. I'm going to go to the questions. Is that all right? We've got 6 minutes left, so there might have to be short-ish answers. I'll let you decide who should take it. The first question is from David Whelan, who asks, is there a link between logophenic primary progressive aphasia and Alzheimer's? My wife of 63 was diagnosed with LPPA about 3 years ago, but the symptoms seem to be moving into other aspects of her capabilities.
Shall I take that one?
Yeah.
So the logophenic variant is a tricky one, and it is one of the language variants of frontotemporal dementia. So there are 3 language variants that we classify, and the logophenic variant is one of them. But the tricky thing with the logophenic variant is that it's often caused by Alzheimer's disease pathology. So pathology, we touched on earlier, is essentially what's happening in the brain itself, and the proteins that are involved in Alzheimer's are different to the proteins that are involved in frontotemporal dementia for the most part. And again, we don't know until we can look at the brain itself what protein's causing the disease. So although it can look very much like frontotemporal dementia, it can look like both. And it's an unanswered question at the moment. The thing that they can do is look at cerebrospinal fluid biomarkers, like Aitana's talked about, for Alzheimer's, and you would know whether it's an Alzheimer's or FTD variant. The one last thing I'll mention is that with all forms of dementia, as the dementia progresses, it does tend to affect other areas of thinking or other areas of cognition. So it could just reflect different areas of the brain being involved, whereas in the early days, only there's one main symptom presenting.
Great, thank you. Next question, Lucy Sharples asks, what kind of postmortem markers are visible in FTD brains?
I can answer that. So it's as we were speaking about the presence of some proteins in the brain. So FTD can be caused by the presence of tau or another type of protein that is TDP-43. And actually, it's not just one type of TDP-43, but you may have up to 5 different types of TDP-43. So it's the actual presence of abnormal protein when you check the brain.
And you can't get those proteins until you've got a brain postmortem.
There's lots of research going on in trying to develop ligands to be able to see them on PET. Yeah, as well as fluid biomarkers, but for now. So for example, in the case of tau, now it's used for Alzheimer's disease, but it cannot discriminate frontotemporal dementia.
Yeah, I understand. That answers that question. Let's moving on to the next one. Rowan De Silva asks, can one reliably select clinical Pick's disease patients for trials for experimental therapy targeting 3-repeat tau?
So that's some nitty-gritty detail on the types of tau protein that we see. And within frontotemporal dementia alone, there are lots of different types of tau involved. and Pick's disease is one group of tau pathology cases, so people with tau protein pathology in the brain. At the moment, again, we don't have markers that specifically tell us which type of tau is involved. However, as Martina mentioned, we're looking at different ways to look at different types of tau using PET imaging, and hopefully as we go forward with fluid biomarkers as well.
And I guess once you've got a therapy, you're going to have to find mechanisms by which to decide who to give that therapy to, and if you haven't, you'll end up giving it to everybody and then seeing who it works in and who it doesn't.
Yeah.
Yeah. All right. Thank you for that question, Rowan. So, I'm going to go to a question on YouTube, which was Lottie Swift says, great to see such an important topic being worked on by such smart people. What led you all to become interested in FTD? Go on, you're going to have to take that one quite quickly because we still have more questions. I'll pick on, does anybody have a particular passion that they want to say, that they want to pick on?
Nobody really knows my answer because I'm pretty sure that's my sister, but if someone else wants to give a go.
I think there are lots of unanswered questions.
It is yours. Yeah, you've got the same surname, right?
I think the number of unanswered questions about FTD and just how diverse it is as a disease, it kind of keeps you wanting to know more.
Yeah,
I understand.
That's why research is so important.
And honestly, you are all inspirational. I think any young people out there that are doing undergrad right now or just finishing at university, we definitely hope that they'd be inspired to consider dementia as their follow-up subject. You don't go working in cancer and other diseases, come work in dementia. That's where we really need you. Alice Sharajda asks, are people with FTD more likely to experience depression and suicidal ideation? If so, what care or support is needed, particularly at that diagnosis? Are they more likely to experience depression?
Yeah, I think with those that are particularly at risk and they know that they're at risk of developing frontotemporal dementia, it can be very challenging. And one of our colleagues, Caroline Greaves, is doing some work around support for those individuals and looking at depression and anxiety within that group. Sometimes with the behavioural variant frontotemporal dementia, it can often look like depression. We don't believe that it's necessarily depression. It could just be the social withdrawal, but there is that overlap there where it may be. And also in those with semantic variant frontotemporal dementia, they often find things very difficult and can become quite anxious and
I understand. And your FTD, this is a good chance to plug your FTD Talk website. Do you want to just quickly do that?
Yes, we've got a new website just launched with loads of fact sheets and support for everyone really, from researchers to individuals with FTD and their families and their carers. So have a look on there if you're interested in learning more about frontotemporal dementia and what we're doing as a lab with our research. We've also got the Rare Dementia Support Group, which is run at UCL, and that covers a variety of different dementias from PCA to FTD and familial FTD. And
Adele Lute makes a good point on the chat as well, which is reminding us that AFTD Awareness Week is this month with rare dementia support taking part. We hope that you're promoting, she's suggesting that she hopes you're promoting this and talking about this on your website as well. And will you have materials that you can share to link to your Just Giving page? Yeah, if anybody wants me to link to that, I can just from our page as well. And just working hard to try and raise awareness of rare dementias and BFTD. And
just one other thing to link to on that note, we're one of our other PhD students has currently got an app out at the moment called Ignite. And that's IGNITE. You can download it from the Apple App Store.
iPad. You've got to have an iPad, haven't you? I did it.
Yeah. So we might, if that's okay, Adam, send you the link. We're looking for healthy volunteers between 20 and 80 to take part. I've got to try and get to 1,000. So
that's right. Somebody's asking if you're using artificial intelligence to look at accessibility of analysis and to I guess look at things like NHS records and stuff like that as well. Anybody doing AI work in your lab? No?
Not really.
Not yet. Maybe if somebody would like to suggest it and put a proposal together, they'd be very interested in
Yeah, very welcome.
AI. And the last question was from Pauline Ross, who says she accompanied her husband to hospital 3 times during his journey through FTD semantic. She found the hospital didn't know or understand FTD. And even though he was finding it hard to express himself, and recently signed a petition for NHS dedicated dementia wards. Do you think this is a good idea? I'm sure you'd all advocate for trying to improve NHS clinician and NHS services awareness of FTD and the symptoms to try and get that more reliably diagnosed, right?
Yeah.
Is there a policy element, somebody in the UCL working on, you know, specifically an FTD in the qualitative sense, like we've looked at with some of the others? I know you're all labs people, but is that something else that another part of UCL is looking at?
I'm sure there must be somebody looking at it. The Rare Dementia Support team look at lots of important things like access to clinical care or care home support for those with dementia, and specifically frontotemporal dementia, as it presents quite differently to some of the other types of dementias. But there's still work to be done for sure. And we just hope that with more awareness in the public, as well as within clinicians of any background, we can get to a diagnosis and one day to treatment quicker.
Thank you, Misha. Did I say that wrong? Right, again.
Misha.
I put a H. Look, I wrote a H. Needs a H in it. Thank you, Misha. Thank you, Martina, Imogen, Aitana, and Lucy. Thank you ever so much for joining us and talking about FTD. If anybody wants to know more, I would highly recommend you go to the FTD Talk website, where there's all information about their research, and please do reach out. You'll find information on all the panelists and their Twitter feeds on the chatfront.uk page that's got information on our researchers. Thank you very much, everybody.
Thank you.
Good job. Thank you.
Thank you.
Bye.